Okay, so here's our vocabulary for our second line of defense of the innate or nonspecific immune system. Phagocytosis and phagocytes and antigen-presenting cells. We've covered these concepts already. We're going to go into a little bit more detail now, but you've seen this before. So the antigen-presenting cells we've covered are just B cells from the adaptive immune system. Now you're going to see some antigen-presenting cells that are part of the innate immune system, and these are called dendritic cells and macrophages. Remember, antigen-presenting cells are special kinds of phagocytes. So let's talk about phagocytes first. So phagocytes are cells that phagocytize, and phagocytosis is a specific kind of endocytosis in which a cell ingests something from the external environment in order to digest it. Phagocytosis is the cellular version of eating. Phagocytes are cells that phagocytize a lot, like as their job. Antigen-presenting cells can phagocytize things, but they also can present antigens of what they phagocytized on their surface in their MHC class 2s, right? Dendritic cells and macrophages are kinds of antigen-presenting cells that are unique to the innate immune system since they do not involve cell memory. B cells are antigen-presenting cells which belong to the adaptive immune system because they do involve generation of cell memory. So when stimulated, dendritic cells and macrophages will release cytokines, which are molecular signals that activate important cellular responses in the immune system. And we have seen cytokines before. So this allows cells to crosstalk between different cells in the innate immune system, different cells in the adaptive immune system, communicate between the innate and adaptive immune system, because cytokines could activate helper T cells. They can activate, we'll get into the cytokines later. Now remember, when B cells were activated, it's not so much that their job is not so much to release cytokines, right? So B cells, when they're activated, their job is to proliferate rapidly, generate more cells with the same immunoglobulin that could attach to that unique pathogen and to generate antibodies. So these are antigen-presenting cells, as are these. So all of them start out the same. They attach to a pathogen, they ingest it, they digest it, and they present fragments of what they digested on the cells on their surface. The difference is what happens after that. So these ones, once they're activated by a helper T cell, they're going to do something different than these. These are going to release cytokines. These are going to proliferate and generate immunoglobulins that fight the pathogen. All right, so phagocytosis again. So remember, you have a bacteria docks with the phagocyte, the phagocyte might be an antigen-presenting cell, but it might not be. And this phagocyte, once the pathogen docks on its cell surface, it's going to bring the cell into what we call a phagosome, which is sort of like a food vacuole. The phagosome is going to fuse with lysosomes, which are these little spheres here. These are digestive organelles, spill out their digestive juices onto the pathogen, digest the pathogen into little tiny pieces, exocytose the debris. All phagocytes can do that part. The part that's unique to antigen-presenting cells is the part with the MHC class 2s. So phagocytes that are also antigen-presenting cells will then display fragments of the pathogen they just digested on their MHC class 2s and wait to become activated by a helper T cell. So there's 3 steps to phagocytosis I want you to know. The first step is chemotaxis and adherence. Chemotaxis we've talked about before when we talked about bacterial flagella. So chemo means chemical, taxis means movement, like a taxi. So phagocytes don't have flagella. They move through more amoeboid-like movement, but they still move towards and away from chemical signals. In particular, they move towards signals, chemical signals from damaged cells and from destroyed microbes. So molecules that are shed by microbes as they divide, as they keep up their cells, those molecules shed by microbes attract phagocytes. And some phagocytes are mobile. The adherence piece has to do with receptor binding on the surface of the white blood cell to some kind of molecule on the surface of the pathogen. So there's a receptor for the pathogen on the white blood cell, on the phagocyte. So you have so the white blood cell is going to move through chemotaxis, not through flagellate chemotaxis, but still through chemotaxis, to movement towards a chemical signal, usually released by the bacteria or by damaged tissue. So the white blood cell moves towards the pathogen, and then it adheres to the pathogen. Step 2 is ingestion. So the white blood cell, the phagocyte, is going to reach its pseudopodia around the around the pathogen and to form a phagosome. Pseudopodia means false foot. So when we talked about amoeboid movement, we talked about pseudopodia. Pseudo means false, podia refers to feet. So pseudopodia are false feet, and so when we were talking about in terms of movement, we say that a white blood cell or an amoeba can reach out a piece of its cell, it extends a piece of its cell membrane and grips a solid surface and pulls the rest of the cell behind it. That little piece of cell membrane that reached out to grab, that's called a pseudopodia. So these pseudopodia are also used to ingest pathogens. So 2 pseudopodia will reach out around like a hug to hug the pathogen and bring it in the phagosome. And then step 3 is digestion. So that's when the phagosome fuses with the lysosome. So those are the 3 stages of phagocytosis I want you to know for the exam. And here's a picture of it. So here's a little bacterium. And so we didn't see the chemotaxis part, but the chemotaxis part was the white blood cell moving towards the bacterium because of some kind of chemical signal. And then once there, you have adhesion. So here are some receptors. It might be a B cell receptor. It might be a different kind of receptor found on other kinds of phagocytes. These receptors bind to some molecule on the pathogen. Then here's the pseudopodia, here and here. Once these receptors bind to the pathogen, these pseudopodia are going to reach around and put this bacterium into a big bear hug, bring it in this phagosome. So that's the ingestion part, that's step 2, and step 3 is when the lysosome fuses. When the lysosome fuses with the phagosome, it's called a phagolysosome, and the material's digested. And then you have exocytosis of the debris. All right, so that's all review. That's easy enough. So now let's talk about the kinds of receptors you might find. So on B cells, you have B cell receptors for pathogens. On dendrocytes and macrophages, you have Toll-like receptors and pattern recognition receptors for pathogens. And Toll-like receptors and pattern recognition receptors on these antigen-presenting cells are going to bind to PAMPs, or pathogen-associated molecular patterns. So Toll-like receptors and pattern recognition receptors are in a way a little bit like B or T cell receptors. They're going to cover the surfaces of phagocytes, and they receive or recognize molecular structures on microbial surfaces. There's 2 differences, though, from B and T cell receptors. The first difference is B and T cell receptors are super specialized. They're specialized to a unique molecule found on a very, very specific species of pathogen. Specific virus, specific species of bacteria, doesn't matter. But their B cell receptors and T cell receptors are specialized and unique to one species of pathogen. Toll-like receptors and pattern recognition receptors are not unique. They recognize microbial molecules that are found across broad ranges of microbes. And I'll give you some examples, but there's something like, one example just off the top of my head is going to be peptidoglycan. It's not just one species that has peptidoglycan. All bacteria have peptidoglycan, right? All bacteria have a cell wall made out of peptidoglycan. And so it's not that all pathogens have peptidoglycan, right? Because some pathogens are viruses, some pathogens are not bacteria, right? Some pathogens are fungi, some pathogens are protists. So it's not that all pathogens have peptidoglycan, but a huge number of them do. It is not a unique molecule, right? So that's the kind of molecule that a Toll-like receptor or a pattern recognition receptor would bind to. And so peptidoglycan is not going to be considered an antigen because it's not going to be antibody generating. It's not there to be bound to by a B cell receptor or a T cell receptor. Instead, we call peptidoglycan a pathogen-associated molecular pattern. A pathogen-associated molecular pattern. These are molecules or molecular patterns that are found over broad swaths of the microbial world. Okay. So let's take a little look at some PAMP or pathogen-associated molecular pattern examples. So down here at the bottom we have peptidoglycan, that's one example, but we also have lipopolysaccharides. Lipopolysaccharides are embedded in the outer membrane. They're part of the outer membrane of gram-negative bacteria. So just to remind you, bacteria have their cell membrane like every cell, and then they have a peptidoglycan cell wall, and then gram-negative bacteria, on top of that, they have an outer membrane. The outer membrane is a lipid bilayer, but it is not a phospholipid bilayer. One side of the outer membrane is made out of phospholipids, but the other side is made out of lipopolysaccharides. So this is also what we call endotoxin. Endotoxin is something very specific. It's lipopolysaccharides, which is it's half of the outer membrane. It's the outer component of the outer membrane of that lipid bilayer of gram-negative bacteria. So it's not that all pathogens have this particular PAMP. Not all pathogens have lipopolysaccharides. Not even all bacteria have lipopolysaccharides, but all gram-negative bacteria have lipopolysaccharides, so it is not unique. Many, many, many species of microbe have lipopolysaccharide, and so it's not an antigen, it's a PAMP. It's a pathogen-associated molecular pattern, and it is not bound to by T cell receptors or B cell receptors. It is bound to by Toll-like receptors and pattern recognition receptors. Another example of a generalized molecule, PAMP, is flagellin. Flagellin is one of the main proteins that builds flagella in bacteria. So again, not all bacteria have this protein, but all bacteria with flagella have this protein. And so it's not unique, it's generalized. So it's for the non-specific, it's not specific, it's non-specific. So it's for the non-specific immune system. It's a PAMP for a toll-like receptor or pattern recognition receptor. And the last example I'm going to give you is lipoteichoic acid. We encountered lipoteichoic acid earlier in chapter 2 when we talked about cell anatomy, or I think it was actually in this textbook, it's chapters 4 and 5. But when we covered cell anatomy, bacterial anatomy, we did encounter lipoteichoic acid, but I didn't make you remember it before. So lipoteichoic acid is part of the gram-positive bacterial cell wall. So gram-positive bacteria, remember, have a really thick cell wall. So just to remind you, so you have gram-positive, so all bacteria have a cell membrane. Gram-negative bacteria have a thin peptidoglycan cell wall and then an outer membrane on top of that made out of lipopolysaccharide. Gram-positive bacteria have a really, really thick cell wall and no outer membrane. And that really, really thick cell wall has these molecules called lipoteichoic acid sticking out of them. So again, all gram-positive bacteria have that. So again, it's not a unique molecule. So if I go back to this slide, we can say that toll-like receptors and pattern recognition receptors are like T cell receptors and B cell receptors, except B cell receptors and T cell receptors are specialized and specific for the specific immune system. Toll-like receptors and pattern recognition receptors are generalized for the they're non-specific for the non-specific immune system. PAMPs are like antigens, except that antigens are unique to individual species of pathogen. PAMPs are generalized, found across many, wide ranges of pathogens. And so antigens are targeted by the specific immune system, the specialized immune system. PAMPs are targeted by the general non-specific immune system. Okay. Another way that toll-like receptors and pattern recognition receptors are not like B cell and T cell receptors is that B cell receptors are found only on B cells. T cell receptors are found only on T cells. Toll-like receptors are found on phagocytes, but not only on phagocytes. They're also found on other kinds of cells in the body. And we'll talk about that when we get to the human microbiome, because the toll-like receptors, for example, in our gut, the cells that line our intestines have toll-like receptors on them, even though they're not immune cells. They're intestinal epithelial cells, but they still have toll-like receptors that communicate with our microbiomes. And so those toll-like receptors on our gut lining are important for programming our relationship with our microbiome. So toll-like receptors are not only found on immune cells. So let's move on to cytokines. This is another review for you. So cytokines are molecular signals that activate important cellular responses in the immune system. They're little chemicals that allow different kinds of immune cells to communicate with each other. So you can think of them in the same class as neurotransmitters or hormones. Neurotransmitters allow different nerve cells to communicate with each other, and hormones allow different organs to communicate with each other. Cytokines are chemicals that allow different immune cells to communicate with each other, and this allows all the different kinds of immune cells we're learning about to cooperate with each other. So you've already learned about cytokines when we covered the adaptive immune system, but there's 3 specific classes of cytokines now that I want you to know. I want you to know interferons, TNF-alpha, which stands for tumor necrosis factor alpha, and interleukins. So there are a great number of cytokines. These are just a few that are going to be important for this class. So interferons interfere with viral multiplication by causing apoptosis of viral infected cells. And we actually saw interferons already when we talked about viruses. So when a virus infects a cell, that cell will release interferons, which are a kind of cytokine, and that interferon will shut down transcription translation, cause death of the host cell. They'll also travel to neighboring host cells to cause death of the neighboring host cells. It's an act of self-sacrifice to contain the viral infection, because if the virus can't replicate inside the neighboring cells, it can't spread, right? So interferons, they shut down transcription translation. So they are specifically to stop viruses from replicating, because bacteria don't use transcription translation, they just eat your cells. Viruses aren't alive, they don't eat, but they copy their genetic material using transcription translation. So interferons stop viruses by stopping transcription translation. The result is the death of the host cell, but also halting the spread of the virus as well. So interferons are one kind of cytokine. Tumor necrosis factor alpha. This has a number of roles. For one, it helps initiate fever and inflammation. And fever and inflammation are parts of our innate or non-specific immune system. So when we are infected by any kind of pathogen, tumor necrosis factor alpha might be released and give us fever and inflammation. And we'll talk at the end of these lectures about how fever and how inflammation helps us fight infection. So they have other roles as well, but those are the roles we're really going to cover. Now, there's quite a diversity of interleukins, each with different roles. Some interleukins will activate immune response, and some interleukins will dampen immune response. Each interleukin is given a unique number. I'm not going to ask you to know the role of specific interleukins now, but I want you to know what interleukins are in general. I want you to know that interleukin is a class of cytokines, because we are going to learn about one kind of interleukin that's very important in immune system training by our microbiomes when we get to the microbiome next week. So I want you to know, so just to recap, I want you to know these 3 cytokines: interferons, tumor necrosis factor alpha, and interleukins. Interferons help stop viruses from spreading through your body. Tumor necrosis factor alpha helps initiate inflammation and fever. Interleukins have many, many roles, some to activate the immune response, some to dampen it, and we're going to really get to one kind of interleukin later when we talk about the microbiome. Natural killer cells. You've also seen natural killer cells before. So again, see, we get the opportunity to review this challenging material a couple times before your final exam, which is why I wanted to start with a harder chapter first. So natural killer cells recognize cells that are not displaying the self-tag, or the MHC class 1s, the major histocompatibility complex class 1s. Natural killer cells target these non-self cells for destruction by poking holes in them and filling them with proteases, which causes the cell to undergo apoptosis. So natural killer cells are looking for MHC class 1s, which are the self-tag. Any cell that does not have the appropriate self-tag is going to be killed by the natural killer cells. The kinds of cells that natural killer cells kill are eukaryotic cells that are not a part of your body, like parasitic worms and fungi. Those eukaryotic cells that are not a part of your body are too big for your white blood cells, which are also eukaryotes, to phagocytize. So eukaryotic pathogens can't be phagocytized because they're too big. So that's a very important role for the natural killer cells. So they will sidle up next to them, punch a hole in them, and spill proteases into them. And then also sometimes natural killer cells can target tumor cells or viral and virus infected cells, because sometimes, not always, but sometimes tumor cells do not display MHC class 1s properly because of rapid mutation rates inside the tumor cells. And sometimes viruses will knock out a cell's ability to make MHC class 1s. So there's some viruses where they won't allow the host cell to have MHC class 1s. In both those cases, the natural killer cell will think that those compromised host cells are foreign cells and kill them. All right, so leukocytes and lymphocytes, these are names for different kinds of white blood cells. Okay, so I want to get into organizing all the kinds of white blood cells, all the kinds of blood cells that we've talked about. So I'm going to stop this video here for now, and in the next video we'll organize all of the different kinds of cells that we've covered, because now you've got the B and T cells from the adaptive immune system, you have phagocytes, you have all these different kinds of antigen presenting cells. How do these all fit together? So that's going to be the next video.